What magnification do you need to do microscopy? Hello and welcome to the Microbehunter Microscopy Podcast. My name is Oliver Kim. Well, let's talk a little bit about microscope magnification and the magnification that you need to observe the things that you're interested in. And when you go microscope shopping, often in catalogs or nowadays more online, you will see a whole range of microscopes being advertised. And one of those parameters that the vendors of microscopes always uses is the magnification, basically. So that means you can buy microscopes sometimes all the way up to a magnification of, let's say, 2,500 times, totally unrealistically high in my view. We'll talk about this later. But the real question is now, what magnification do I need? Or is 1,000 times already enough? Or can I already see, I don't know, ciliates, water life, bacteria with, let's say, 400 times? Or what do I need? Okay. So basically the point of this podcast episode here is to provide some clarity, and I can already comfort you a little bit in the sense that it's actually much easier than it might appear, because when you go out and buy yourself a microscope, and it doesn't really matter which compound microscope that you buy, if it magnifies all the way up to 400 times, you are already covered, and you can see, I would say, maybe 95% of the things that you probably ever want to see. Okay, so 400 times total magnification is already enough. If you want a little more than 600 times total magnification is even a little bit better. It's a little bit more difficult to use under certain circumstances, because the distance between the objective and the cover glass of the slide is very, very small. So it's a little bit mechanically, it's a little bit delicate in that sense, but generally pretty much you're covered. Okay, so I would say that when going out and shopping for a microscope, I would say that magnification is one of the least important parameters, which is a little bit ironic, I have to admit, because isn't this the point of a microscope, kind of to make small things larger, and then all of a sudden I'm saying here it's actually one of the least important ones. And the reason is why I'm saying this is because the microscopes already cover such a wide range of magnifications that you're covered already right from the beginning anyway. But especially beginners, what they do is they often ask me, they say, okay, I want to see, I don't know, a cell, let's say a plant cell. Can I see that with this particular microscope? And the answer is I don't even have to look at the microscope. I can already tell you right in advance yes, okay? Because cells are already quite large anyway. Most cells, except bacteria, most eukaryotic cells are quite large anyway, and you can already see them quite well, at least most of them, even at surprisingly low magnifications. Okay. It really depends a little bit what your interests are and whether you want to take pictures, because if you want to take pictures through the microscope, then some objects, of course, you want to have a little bit more flexibility in magnifying the things that you look at. But let's now have a more detailed look at the objectives that come with a microscope. And when you buy a compound microscope, so I'm not talking about stereo microscopes now, which are a different thing, but conventional compound microscopes, when you basically buy one of these, then most of them the basic models come with 3 objectives, and they magnify 4 times, 10 times, and 40 times. That's kind of a little bit of standard. Some better or more expensive microscopes, they might add a 100 times oil immersion objective, so they have a total of 4, or a 60 times non-oil immersion objective. That's also quite possible. So this basically gives you now a total magnification of 4 times 10, because 4 times 10, because the eyepiece that you look through, this is basically all you have to include this into the calculation. It gives you a total 4 times 10 gives you a total of 40 times magnification. Then the 10 times objective with the 10 times eyepiece, 10 times 10 is 100, of course, and then you have the highest magnification that you can then get is 400 times with 40 times 10. Okay, so what you do is you always multiply the magnification of the objective with the magnification of the eyepiece. Now, if you want to have a higher magnification, then you can either buy yourself a better, or not better, but a more expensive objective that magnifies more. That is actually the way that you should do it, okay, because this way you also preserve the resolution. You get a clearer image. But the easiest and simplest way to increase magnification would simply be to exchange the eyepiece, okay? So if you exchange the eyepiece for a 10 times eyepiece with a 20 times eyepiece, of course you're doubling the magnification again. So you can, with a 40 times objective and a 20 times eyepiece, you get a total magnification of 800 times. This might be useful in certain circumstances, but you have to be aware that this way you actually lose quite a little bit of resolution as well, because it's actually the objective that determines the resolution and the clarity and the sharpness of the image. So, but generally, if you want to look at water life, for example, you go. That's one of the most common things in amateur microscopy. You go out and you collect some water samples, some algae, maybe some yucky stuff from the bottom of the pond that you want to look at. And those microorganisms that you find in there are actually quite large, and they're too small to be seen properly without a microscope, obviously. But with a microscope, they're quite surprisingly large, actually. And you do not need a lot more magnification than around 100 times or even 200 times. So what I do is I have in my microscope that I have, is I bought myself a 20 times objective. It's not a very common one, because it's not, but it, for water life, 20 times objective with a 10 times eyepiece gives me a total magnification of 200 times, and that's really nice because most of the water life can be seen sufficiently large, but not too large, so I can basically easily also follow them around, because if you magnify very much, like 400 times or even more, then the water organisms that move around, they move away out of the field of view so quickly that you actually lose them, and then it's very difficult to actually chase them. So especially for fast-moving water organisms, you actually want to work with a low magnification. So I often use only the 10 times or 20 times objective. And if you already want to really make them large, because you actually want to look into the organism, because many of them are transparent, so you can actually look into the organism and look at a little bit at the stuff that's going on in them, then I usually use my 40 times magnifying objective with a total magnification of 400 times. So you see, you're covered, okay? So the question is now, when would you actually need a 100 times oil immersion objective? Are there any cases? Well, first of all, yes, there are specific cases, specifically if you want to look at, let's say, chromosomes, or if you want to look at bacteria, and if you want to see them a little bit larger, then I would recommend a 100 times oil immersion objective. However, I'm only recommended with reservations, because there are certain disadvantages as well. I would say that for in most cases, the oil immersion objective might cause, if you're inexperienced, might cause more problems than it resolves, because with the oil immersion objective, what you have to do is it comes along with so-called immersion oil, and what you have to do is you have to put a drop of immersion oil on the microscope slide, and then you have to rotate the oil immersion objective into the oil. This means that the bottom lens of the objective becomes covered with oil, and you do that so that there is no air between the lens of the objective and the specimen slide, the microscope slide, and because this increases brightness and resolution. So basically, these objectives are made in such a way that they're oil tight, so that you can actually cover them in oil, the bottom part. And this, however, has also a disadvantage. You got to clean the objective. Once the slide is covered in oil, you cannot use the other objectives, the non-oil objectives, with the slide, because the slide is covered in oil, and you do not want to get any oil on the non-oil immersion objectives either, because they're not designed to do that, and then it really messes them up. So you see, only for experienced people, and probably not even in educational use. We use them because the students, they confuse the objectives and so on, and yeah. So I think I'm getting a little bit sidetracked now. I just wanted to show you a little bit that if you want to work with very high magnifications, then this only works with specific objectives, okay? and that there are some disadvantages with that. However, I would say in most cases not needed, and larger bacteria, especially bacteria that you can find also in water samples and so on, can also be seen quite well with the total magnification of 400 times. Okay, there are other parameters really that reduce the visibility, like lack of contrast and so on, but it's not really magnification. Okay, so so what am I basically what's the take-home message? The take-home message is go microscope shopping, and don't worry about the magnification, the maximum magnification too much, because it covers the whole range already, and look for other factors that you want to have. For example, are you able to attach a camera to the microscope? I think it's much more important, for me at least, than to have a very, very high magnification, which I'm not using anyway. Are you able to attach a camera to it? Is the microscope sufficiently stable? For example, I like large and heavy microscopes, okay, because this gives me more stability, but of course at the same time you cannot carry them around as easily, which might also be important sometimes. Maybe light intensity could be another factor, especially if you want to take pictures. Is it basically a monocular or binocular? Basically, do you look through it with one eye, or do you want to look through it with two eyes? So there are other factors that I would actually worry about, and not so much magnification. I kind of compare the whole issue with magnification a little bit with the regular photo cameras. It's a little bit like asking, if you want to buy yourself a camera, it's almost like asking, well, which zoom lens in my camera do I need so that I can take pictures of my cat? Okay, with all of the cameras you can take pictures of your cat. There are other things that are important. I mean, I don't know, the intensity of the flashlight maybe, or how heavy the camera is, or the resolution maybe, of the sensor. You see, but it's not really like this, that the optics are limiting in any way. Okay, so that is kind of the analogy that I sometimes would like to give you. So basically, if you go microscope shopping, you can be a little bit relaxed, and as long as your microscope has 4 times, 10 times, and 40 times, you're already covered with 95% of the cases. Get yourself a 60 times objective for high magnification, and if you know what you're doing, and if you really go a little bit more advanced, get yourself a 100 times oil immersion objective. And if you want to do a lot of water, if you want to look at a lot of water organisms, then I found the 20 times objective also very meaningful and useful. However, they're not quite as common, so you simply have to look around a little bit. Okay, so this was basically it already. I wish you all the best. Happy microbe hunting as always, and bye-bye. See you next time.